Tuberculosis, which is caused by Mycobacterium tuberculosis, kills between 1.5 to 2 million people a year. So just to put that in perspective, that's about the size of Indianapolis every year. So that's a huge number of people. The number of cases of TB has actually risen from 1980 to now. Mycobacteria are not only pathogenic and live inside of a host like us, but they live inside cells. They live inside a type of cell called a macrophage. And so we were interested in how they invade, how they survive within those cells, and how that relates to the eventual disease state that you see both in animals and in humans. I had a very talented graduate student in the lab, Sanchita Bhatnagar. She, of course, graduated and left, as good students often do. When she was here, she made the, I guess, serendipitous type of discovery. And you know, many things in science work that way. You don't expect something to happen, and all of a sudden it does, and it leads you in a whole new direction. And that's sort of what happened in this case. We were interested in a glycolipid. It's called GPL, glycopeptidolipid. And part of those studies, we wanted to see where the GPL goes. Is it released from the mycobacteria during the course of infection? If it is, where does it go inside the host cell, the macrophage? But what we found very surprising was that we started seeing these GPLs, these glycopeptidolipids, outside of the infected cell. We started seeing them in cells that were not infected. So it's like, you know, what's going on here? How does this happen? And to make a long story short, I did a lot of control experiments and things that you have to do, especially when you find something unexpected. Glycopeptidolipids were packaged in small little vesicles that are released from infected macrophages. They then travel to another cell, and in that second cell, that uninfected cell, can take up these small little vesicles, these membrane-bound vesicles. And these vesicles have a name. They're called exosomes. When we isolated these exosomes from infected cells, we identified 41 different mycobacteria proteins. If they have mycobacteria proteins, that's pretty diagnostic of being infected with mycobacteria. TB is a lung disease, right? It's a pulmonary disease. So TB primarily a lung disease, so it's found in the lung. So these exosomes are released from infected macrophages in the lung, but they don't stay in the lung. They actually get into many different bodily fluids. They get into the serum or the blood, they get into saliva, they can get into urine, breast milk. I mean, all these different types of bodily fluids have exosomes. So, you know, we can use the fact that the exosomes get out of where the infection is happening and disseminate through the body. Okay? Our goal with this is to find those mycobacteria proteins in a large number of patients and see if this is a good biomarker. What the Gates wants, the Gates Foundation and many people in the TB field want, is a good TB diagnostic that can be done at point of care. Meaning you can go to the villages, you can test those individuals and say, do you have TB, do you not have TB? And you want to be able to do it within a couple hours. Okay? So that's what we're looking for. And if we could do that, then it's estimated we could save about 450,000 lives a year if we had a TB diagnostics that was 85% sensitive and 95% specific. So that's a large number of people, because it turns out only about 60% of the people who have TB are accurately diagnosed. So you can have the best drugs in the world to treat these, but if you don't diagnose them, you know, you're not going to be able to treat them, and you're missing a lot of people who die. So that's why diagnostic has become such a big issue, and why the Gates Foundation is putting an emphasis on it.